Hello students, this is Dr. Faidan Mirza. I am discussing questions related to selection and evolution with you in this video. Okay, so this question came in March 2018, variant 42. The question actually deals with a red deer. They are saying that it's a herbivore and that it feed on wide range of plants. It says that in UK, the number increased from between 1960 to 2010, and the table shows the details to you. You can see all the values I mentioned to you. They are saying calculate the percentage increase from 1960 to 2010. So when you are calculating percentage increase, you have to make sure which years or which values they want. So they want you to calculate the increase from 1960 to 2010. So what we'll do, we take out the final value, which is 360,000, and we'll use the initial, which was 135,000, and we have these two values here. We'll subtract the final value from the initial value, and then we'll divide it with the initial value and we'll multiply it by 100. So this will give us the answer. Then they are saying the answer should be in one decimal place. So 166 point, this value need to be rounded off. So our answer is 166.7. Then the next part is a bit simple. They are saying environmental factors that affect the population size of red deer so that numbers do not continue to increase forever. Suggest environmental factors that may prevent further increase of the size of the red deer population. So what are the controlling factors in the environment? So yes, loss of habitat for grazing, shelter, it can lead to an increased vulnerability towards predation. Plus, you can say that, well, it will result in reduced food supply. And when the food supply is reduced, there would be an increased competition for the limited food sources available. Diseases may limit the size of population of this deer as well. The next part, they are saying the body mass of red deer shows wide variation within populations. Of this variation is shown to you. This is low, medium body mass, and high body mass. The selection pressure acted consistently over many years against red deer of low body mass. And if it's low body mass, we know that the low body mass will cease to exist. And if it ceases to exist, that means the graph will shift a bit ahead. So we need to show directional selection in this graph. They are saying that what you need to do, you need to draw the graph of directional selection. So what we need to make sure we just make a similar graph of this sort, but now the graph will be shifted ahead from the dashed line because the mean will be redefined. Initially, the mean body mass, the medium body mass was most frequent in population. Now, not the medium, but somewhere between medium and high, this body mass is most frequent in the population because the low body mass got selected against. The force of selection will be directional selection in this. Further, they are saying selection pressure acted consistently over many years against red deer of medium body mass. So if medium body mass goes out, so we know that this is the medium body mass, this goes out. So the graph will have two peaks here. And this is the kind of graph that we'll be making because it's disruptive selection. So it will have two peaks and both peaks will actually have the same amplitude and same height, and they will be much lower than the actual peak that was initially there. And this time the force of selection will be directional selection. Oh, sorry, disruptive selection, because the directional selection is when the allele when a different phenotype is being favored, whereas the disruptive selection is when the actual mean which was there in population, the central value, this starts to get selected again. So this is disruptive selection. It disrupts the normal distribution into two different size of population, and both are almost in equal abundance, and this is disruptive selection. This is an example of disruptive selection. So here they are saying this is March 2019, variant 42. This question came for these toads, and they are saying these Mexican toads they live on land, but they go to pond as well. Their eggs are laid in water. They hatch into tadpoles, which feed on ponds before turning to adults. They are further saying that these are of three types. There are omnivore type of tadpoles. There are carnivore type tadpoles as well. Not three types, two main types. So there are carnivore type and carnivore type. These are the two types of toads here. The difference between phenotypes of these two tadpoles are related to their different feeding behaviors. Now, what is the omnivore type doing? Omnivore type is feeding on tiny pieces of detritus. What is detritus? Detritus is the dead material from the plant and animals. And algae, it basically is feeding on two things: microscopic photosynthetic organisms, which are algae and detritus, at the bottom of the ponds. So, and they grow slowly. So we know the omnivore type. These features are already mentioned to us because this information is very relevant for whatever we need answering further. The carnivore type tadpoles feed on small animals in water, such as fairy shrimp, and small omnivore tadpoles. The tadpoles grow quickly. Now they are shown to you here. This is the fairy shrimp, this is the omnivore type tadpole, this is the carnivore type tadpole. So these are the two types which are actually mentioned to you in the diagram, in the description above. So this one is the omnivore, and this is the carnivore. Now, keeping this information in mind, between these two main types of tadpole, there's a continuous range of tadpole with intermediate body phenotypes and feeding behaviors. It means it's a continuous variation. For any tadpole, regardless of age, it's possible to calculate a phenotype score depending on features of the tadpole. A tadpole with phenotype score close to +3 is a typical omnivore. So plus a value of 3 here is omnivore, a value close to 7 is a carnivore. So we know that from this diagram, it means that from this description that a typical carnivore will be 7 and a typical omnivore will be 3. So these are the scores given to us according to the description which the question is giving to us. The phenotype scores were determined for a large number of tadpoles sampled in 2 ponds. Further, they are saying the availability of detritus algae was high for one pond and low for the other pond. All other conditions of the 2 ponds were similar. For both ponds, phenotype scores were determined shortly after the tadpoles had hatched from the eggs and 10 days later. Now you can see this in the diagrams, figure 4.2, that how this actually is happening. You can see this disruptive selection occurring here. And you can see that 3 and 7, they are less, whereas the intermediates are very high throughout. But 10 days later here in this, you can say that disruptive selection has occurred. So what are they saying? Yeah, they are asking using results shown in figure 4.2, describes and suggests explanation for the change in the frequency of tadpoles in the pond with low availability of detritus and algae. So low availability of detritus and algae, it means that we have to refer to this curve here, the curves for the low availability. You can say that in this condition, disruptive selection occurred, and they both the score 3 and the score 7, they increase, where the intermediates they decrease. So this is what we're mentioning. The frequency of omnivore and carnivore, they remain high. The extremes of the phenotypes remain more frequent than the intermediate. This is an example of disruptive selection. The food availability, we can obviously, if the food availability is a selection pressure here, because it's in this case, omnivore and carnivore had a selective advantage. They consume food resources more readily than other intermediates and are less likely to survive and reproduce due to low availability of the food. So further, they are saying 11 days after the eggs hatch, the mean body mass of omnivore tadpole and carnivore tadpoles in the pond of low availability of detritus and algae were measured. The results are shown in figure 4.3. So you can see the data here. The bars here need to be seen which value they are showing. So this bar is actually of 0.58. This bar is about 1.1. You can just read it from the y-axis here. They are asking you to calculate the mean body mass of omnivore type as the percentage of mean body mass of carnivore type. So what we'll take, we'll just take the value of 0.58 and we'll represent it with 1.1 because this is what they are asking us. And we'll multiply by 100. The answer comes here in decimal. We round it off to a whole number. The answer is 53. The last part of this question goes as they're saying that in a different pond with very low availability of detritus and algae, all of the conditions were the same as the first 2 ponds. As previously, measurements were taken shortly after the eggs had hatched, and 10 days later, after 10 days, nearly all the tadpoles in this pond were carnivore types. So you can see this is an example of directional selection. Now you can see that this is shifting ahead. So what's happening here? They are asking you that which force is acting. So it's directional selection occurring. Suggests explanation for the change in the phenotype frequency of the tadpole with very low availability of detritus. Selection pressure works against the omnivore type. Omnivores die as food sources run out. They get eaten by carnivore type. Furthermore, the carnivore type increase in pond, its area frequency increase, and it would increase in population. Last, towards the end, they are asking, although they are very short questions, but it's a very lengthy question with a lot of description. So you need to make sure that you carry on all the information which is given to you in this question to answer all these subparts. They are saying that Mexican tadpoles develop into adult toads that do not live in water. In some years, the pond of Mexican toads tadpoles they dry out quickly. Suggests why carnivore type tadpoles have a selective advantage in years when the ponds dry, they become dry. Basically, they grow faster into adults and they grow in shorter span of time. And this information was also given to us early in the question. This question from major in 2019, variant 43, they gave three theories here. Basically, they are saying regressive evolution. This is a new concept that they have given in this question. But although you already know information regarding the theory of evolution, so it's not that difficult, but it's a different question. So they say that regressive evolution is a change in population over time that involves loss of certain phenotypic characters. So yes, certain features get lost over time. It is thought to be caused by genetic drift or natural selection. You can recall what genetic drift and what natural selection is. An example of regressive evolution is the loss of eyes in one of the Mexican cave fish. The name is given to you. The eyeless cave fish live in caves that are in total darkness. So if they are in total darkness, they would not need eyes to begin with. But how have they evolved? There are three theories to explain how loss of eyes in cavefish has occurred. The theory A is there is no advantage of having eyes in the cave that is in total darkness, where energy sources are scarce. Having eyes is a disadvantage that may lead to energy cost. So yes, that's a possibility. Theory B, a mutation has occurred in a single gene. The mutation has two effects: a lack of eye development and increase the number of chemoreceptors on the skin. When chemoreceptors are more present more on skin, so this kind of fish will be more receptive to its environment. It will be responding a lot to the chemicals present in its surroundings. So lack of eye can be accommodated by these chemoreceptors because eyes actually catch the information from our surroundings. So yes, chemicals can be sensed by the skin, and they may be sending the same kind of information. So we can relate because bats are blind. Bats use sound waves, and the sound waves that are reflected back to them, they calculate distances according to that. So that's just some basic information from your old general science classes, or some information that probably you must already be having. Anyhow, coming back to theory C. Theory C, various mutations occurred in genes responsible for eye development over a period of time. Over a period of time, by chance, these mutations increase in frequency in small isolated population. Eventually, this produces a population of eyeless cavefish. So there are three different situations given to you. State one theory ABC will describe genetic drift. So genetic drift definitely refers to a small isolated population. So in the first two, there is no reference to small isolated population. So we'll mention theory C. Is going to explain which theory or theories are based on natural selection. So theory A and B are based on the natural selection because they both are based on natural selection, energy, excess selection pressure, producing eyes as a wasteful of energy. Less energy will be required by eyeless fish, so they can survive in limited food supply. They have more chemoreceptors on the skin to assist the detection. Over the time allele frequency of this phenotype increase, so reproduction allowed favorable traits to allows to pass on to the next generation. There are several separate populations of eyeless cavefish. These populations spend time in areas where with light, and fish in these populations have eyes. The mitochondrial DNA of eyeless were compared with mitochondrial DNA of with eyes, suggests how DNA evidence can help find out whether the eyeless fish and the Mexican this eyes with eyes the same species or are the different species. So how this can be compared? We know the difference in the base of the DNA sequence of the nucleotides can be counted and compared. If the mutations are less, this means they share a common ancestor in recent past. And we can do this with microarray and using bioinformatics as well. So just why mitochondrial DNA is used instead of nuclear DNA for studying closeness of relationship between two populations. We know that mitochondria basically is having a maternal origin of the DNA. There is a maternal lineage. There is no crossing over occurring. Mutation rate is pretty constant. The DNA is not read by histone proteins. It is easy for analysis because there is a less number of genes. Plus, there are many copies of mitochondrial DNA in a cell because it's not just one mitochondria. There are many mitochondria in a cell, so every mitochondria will have the mitochondrial DNA, unlike the nuclear DNA, which is just present inside the nucleus, and one cell will be having one nucleus only. That's it from my side. I hope that was helpful for you. Thank you so much.